if you had to pick, this is a question we ask of many people. If you had to pick a dead scientist to bring back and to sit down and have a beer with, who would you pick and what would you talk about?
Oh, it'd have to be George Bartholomew.
Yay!
Good choice.
I mean, yeah, he would be, the grandfather of comparative physiology, sort of all sorts of interesting topics to talk about. So,
yeah. So what do you think he would think about modern biology?
He'd probably be disappointed that a lot of the old stuff has gone missing. You review papers these days, and science started in 2000.
That long ago? I thought it was
George
2010.
Bartholomew. Who's this guy?
So yes, I think that would be a little bit disappointing. There's so many things that have been discovered that's like, no, this was actually done in the 50s, 60s. So yes, I think he might be a bit disappointed in that, but I think he'd be really, impressed that his tree has grown dramatically. I mean, so many of us are a branch on the tree or a branch on a tree. So
yeah, be fun to have him sitting there in the audience for the Bartholomew Award at the SICB conference.
Yep.
Do you think there's a technique that's come out since he passed, or a study area that he'd be particularly excited or upset about? I mean, you sort of alluded to the fact that we don't talk about the old literature, so maybe we've covered that area. But is there something that something specific he'd go for?
Probably just the opportunities that some of the new technology now provide. So, you can ask the same sorts of questions, but you now have fantastic opportunities to, have smaller and smaller data loggers and implants and some of that technology, GPS technology. So that, I think that opens up lots of opportunities for us to ask more questions or to get more precise data on old questions.
Yeah. Okay. So switching gears a little bit, tell us about a non-science hobby that you have.
Photography.
I'm
a very keen but very poor photographer, but I really enjoy it.
Awesome. What are your subjects?
Animals.
So biology next to that. Have you had any of your photos show up on journal covers or anything like that?
Yeah, I get the odd journal cover, couple of Ecological Society calendar inserts, but that's about it. I'm not real good at it, but I enjoy it.
It's much better than I'd be.
And actually, I enjoy the process probably more than the result. So it gives you a good excuse to sit there and hang out with an animal for 2 hours. So you might sit there with an echidna for 2 hours taking photos, whereas normally you look at it and go, oh, that's nice, and move on. So I think I enjoy the process more.
Yeah. So you're one of those people out there with a giant long lens?
No, I have a lot of lens envy. So
lens envy, yeah,
a cheap lens. So
I'm into photography myself, and I've been like drooling over lots of equipment recently, and it would just completely break the bank if I went for it. So I'm not going to do it.
Yeah.
Is there a particular photo that you're particularly proud of? I mean, it could be the outcome, but it could also be, like you say, the process of what you had to go through to get it, or
not really one in particular. So it's a mix, because some of the better photos are not the ones that you worked the hardest for or went to the most exciting place. And the really frustrating thing is that my husband, who usually carries my spare camera that I'm not using at the time, gets bored and starts snapping pictures and takes a better picture than I did. So most of the best photos that we have are the ones that he's taken while he's been bored and just playing with my spare camera that he's holding for me.
Great.
Which is pretty funny.
It's your camera. I think it counts. I think you could get some credit for that.
He's like, here, hold this while I use this, and he'll be like, oh, click, 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 and it's like, oh.
Good. Well, should we move on to the main thing, Marty? You want to?
Yeah, let's do it. All right. So thank you so much, Christine, for joining us today, especially given the time difference among the three of us. And Art, I guess you're up pretty early to make this happen too. So I'm the only one with a reasonable time, although my coffee fill is a bit on the low side. So we'll see what we can do there. We want to talk about a lot of things today. We're going to start in the arena of climate change, and especially the heat waves that we experience a lot of parts of the world now, but particularly where you are in Australia. You studied a lot of things in the last few years, including the iconic species of Australia, like the echidna. But you've particularly done a lot of work on birds. And so let's set up where we're going, talking about what we know about climate change effects on birds in Australia, and in particular with regard to these heat waves.
Okay, so one of the things that we've noticed in Australia is that there's an obvious increase in mean climate, but one of the more probably pressing things for wildlife is the potential for an increase in extreme events. So higher maximum and minimum temperatures, and these can be more problematic. So for terrestrial animals, they already deal with pretty big diurnal and seasonal differences in climate, so they can usually accommodate small mean increases, but it's these extreme events that can become much more problematic. And birds in particular can be susceptible to these because they're generally small, they have a poor ability to store resources, they're mostly diurnal, they don't usually shelter underground or places like that like mammals do, so they really take the full brunt of some of these extremes. And particularly in Australia, we have seen lots of examples of mass die-offs of birds, where hundreds to thousands of individuals have been impacted by some of these extreme heat events. So it makes a really interesting system to study.
Mm-hmm.
I've seen pictures in the last few years of, you know, a carpet of dead birds, and it's just really, really shocking.
Is it the maximum temperature on a given day that leads to mortality, or is that the temperatures are sustained for a few days, or both?
I think it's both. And one of the things that we found is that birds seem to cope pretty well with really high temperatures. And we think that it might be one of two things. So when they can't predict extreme weather events, so if really high temperatures comes out of the blue and they can't necessarily prepare for that. And sometimes when it's really extreme and it's really prolonged and the minimum temperatures are high and they just don't have the time to forage and drink during the cooler parts of the day. So I think it's really extreme temperatures for a long period of time, or really unpredictable extreme temperatures that just come out of the blue.
Okay. And these extremes, I mean, you're sort of hinting that in the past they have happened. It's that they're a lot more common than they used to be. But when we call it extreme, like that's a relative comparison. So what is that? I mean, is it, you know, surface? It's definitely not surface of the sun hot, but how does
almost
the heat almost depends on where you are in Australia, right? But how hot is it compared to, you know, so the average hot day?
So the Bureau of Meteorology in Australia defines a heat wave as 3 consecutive days with minimum and maximum temperatures higher than the typical temperatures for that area at that time of year. So it obviously really varies depending where you are. But some areas, you know, you're looking at 46, 47, 48 degrees Celsius. So, you know, we've just had, you know, a week ago, another round of, you know, record temperatures where we were getting 47 degrees in some of the inland parts. So
it's so hard to imagine. Yeah. Well, let's talk about that. So where in Australia are these problems the worst? I mean, you know, it sounds like the biota in any particular area must be, you know, more or less adapted to that area, and so they may be experiencing heat waves everywhere. But where are the heat problems the worst?
Probably the climate change issues are most seen in our alpine regions. So we do have alpine areas in Australia, but they're relatively low in altitude. So it's about 2,200 metres is the highest point on the continent. And so for species there with climate change, there's just nowhere for them to go. So we're getting less snowfall, earlier snow melt, and things like mountain pygmy possums are in real trouble because they hibernate, and they just, you know, they come out of hibernation too early, so their food hasn't arrived, and so there's this big mismatch. The other areas that are probably most susceptible are some of the highland rainforest areas in North Queensland, because again, they're sort of high altitude, nowhere else to go. And so they're probably the two sort of ecosystems that are most at threat. But the interesting biological ones are the arid zones, because that's already hot and dry and extreme, and they get more extreme. So when we're trying to look at adaptations of species to extreme conditions, then those are going to be the most interesting physiologically.
These alpine regions are really interesting. So I've thought a lot about alpine regions here where I am in Montana, and we're working on some high elevation species and considering this problem of, you know, as climates change, these things could, in theory, move uphill, but there has to be room uphill to go, right? And it sounds like there's not very much uphill room in Australia.
No. So a lot of species, there's just nowhere else for them to go. And I mean, mountain pygmy possums are really interesting because with global warming and snow melt, they actually become too cold. So they actually hibernate underneath the snow. So they'll be sitting there at, you know, a degree, and they can hibernate and they don't have to thermoregulate because they just thermoconform at a degree while they're hibernating. But if the snow melts and it gets down to -5 degrees, they suddenly have to thermoregulate. So it shortens their hibernation period. And when the snow melts and it gets warm in the spring, then they come out of hibernation, but they rely on bogong moths, which move to that area in the spring that they feed on. But if they come out of hibernation a month before their bogong moths arrive, there's nothing for them to eat. So they actually have the problem that it's actually too warm, and that is not good for them. So it's quite an interesting situation with these guys.
Interesting.
Uh-huh. Looked like you were going to ask another question. So I'm going to come back to this predictability, unpredictability issue that you mentioned. What's a consensus right now in the field? I mean, maybe as regards, metabolism, which is going to be the focus today, but if you want to take it more broadly, that's fine. What's a consensus now about the extremeness of an environment in terms of sort of the averages versus the variation, and especially the unpredictable variation? Is there a consensus on which one of those conditions is more challenging for organisms?
Yeah, it's probably the extremes and the unpredictable extremes that are the most of the problem. So, and if you superimpose those on a gradual warming on the top of that, then it becomes even more challenging. But yes, the consensus seems to be that it's unpredictable, really extreme conditions that are the most problematic.
Okay.
So if you project out models into the future in Australia, I mean, what's the expectation for the severity of these events, in 40 or 50 years, and do you expect big shifts in species ranges, that kind of thing?
Yeah, so the modeling obviously depends on best case scenario, but the prediction is that extreme conditions are only going to be more widespread, they're going to be more extreme, and they're going to be more frequent.
Yeah. All right.
No. Yeah, I think. Oh, go ahead, Art.
Nope. Go ahead. It's all you, Marty.
Well, I think what Art was talking about, I'm going to put words in your mouth, Art, is that with the models, these multiple different factors, extreme can only get so extreme, right? I mean, at some point, is the expectation? Well, I'm trying to get my head around whether there's some differences, some reason to expect that certain species that had the potential to deal with one version or the other are going to be the ones that we would expect to be the most common in the future. So if we want to just make it simple, dichotomize that species are either good at dealing with really, really bad but constant conditions, and other species are pretty good at dealing with just variable conditions, which also includes the bad conditions. I mean, maybe that's an oversimplification. Maybe it's just not even possible. But if there's anything like that has some grounding, how should we think about what the future is going to hold? We always bring in our own research systems when we think about this. I work on garbage species and invasive species. And so in cities and modified habitats, there are particular types of species that dominate those places and not others. So in the context of temperature in particular, is there an equivalent? And again, is there this sort of feeling about who's going to be common in the future?
I think a lot of it is more complicated than their physiology dealing with the climate. I think it's the ecological interactions. So a lot of species can probably deal with certain climate factors, but if the plants that they feed on aren't there anymore, then their food's not going to be there. If they require snow, then they're not going to be there. So certain species, it's not necessarily what they can physiologically tolerate, it's whether that their ecology is going to allow them to be there. And there's going to be winners, there's going to be losers. Things are going to be able to shift in some cases, but the species that are really going to be in trouble are those that are really endemic and they've got really small ranges. And then they're the ones that can be hit by these extreme events. So obviously heat waves, but things, fires. I mean, that's the other really topical thing at the moment is bushfires. So if the reserve system doesn't have enough redundancy in it to have species spread across an area, that's when these extreme events can really have an issue. So really endemic species that can't move around. Those with really broad distributions are probably going to be okay because they can move somewhere else. A lot of our birds, for example, are really nomadic. They move around all the time anyway. But it's those really endemic species with really tiny reserves. They require, really long unburnt habitat in a really small area that can really struggle because there isn't anywhere to go. And they're the ones that are going to require really careful management moving forward.
Okay.
I've thought some about evolutionary aspects of this. And I think one really interesting question is how do you think about extreme events in the past and how those might have shaped the evolution of physiological systems and the ecological composition of particular communities? And it's hard to think about that, right? Because it's hard to know how far back in time you have to go in order to get the relevant time scale, right? So like I'm doing a study right now on aspens and leaf mining caterpillars, and we've used some local weather station data to go back 20 years to estimate how often extreme heat events have happened. But maybe the relevant time scale is 1,000 years or 10,000 years, and that's the sort of scale that is relevant to the evolution of these physiological systems. So how do you hindcat? How do you look back into the past and estimate what's happened to shape the evolution of the systems?
I think where you've got big areas of continuous populations, then extreme events that knock off a whole lot of individuals aren't such an issue, because species are just going to move back in there. So there's probably not a huge lot of selection pressure in a natural system for particularly widely distributed species, because there's always going to be movement back in. So you have climate refuges and things like that. It's really when you have this really human modified system where you don't have habitat continuity, that you have a small number of individuals of a population just in one area. And if there's an extreme heat event in that area and it knocks off a whole lot of individuals, you're not going to have new individuals re-moving in. So and you can end up with these sort of population bottlenecks occurring. Because of these. So I think it's a combination of not just the extreme events, but the extreme events on top of changes in land use and habitat and introduced species and all of those things combining together that's made it more of an issue than probably what it was in the past.
Yeah, got it. Super interesting.
You would, I mean, our again, I'm going to second time, and we've been talking for 10 minutes, put words in your mouth. There are parts of the world, there have been parts of the world where the climate is more unpredictable. Right. And so there has been selection for, some sorts of traits that are going to be able to cope with that. So sure, I mean, the endemics maybe didn't experience that, or lots and lots of species wouldn't have had ancestry that included species with those traits, but then there are some that would be. And I think that kind of thing is what I was talking about before. If we know anything about the types of trait constellations in species that may facilitate their success in the future, if they did have this kind of history of lots of extreme unpredictability, the expectation, I think, would be that those would be the ones that could do best. Maybe not with regard to temperature necessarily, but in general.
Yeah, I mean, lots of Australian birds, I mean, their breeding system, for example, is they're not strongly seasonal breeders. They breed in response to good conditions. So they've obviously been selected for not having a predictable environment that, every winter it gets cold and every summer it gets warm and you breed in the spring. They breed when conditions are good. They're really nomadic. So we don't have, very many species that actually seasonally migrate in a predictable way. So they're nomadic. They just move to wherever the conditions are good at the time. So there clearly has been selection for those sorts of traits to deal with that unpredictable and not in a quite extreme environment.
Yeah. Great. Well, let's move on to a somewhat different topic, and this will just sort of segue into one of your main things, which is work on endotherms and the relationship between endotherms and climate and the heat balance of endotherms. And just to set the stage, I would say, I view it as sort of two main ways that organisms can regulate body temperature, right? So if you're an endotherm, you're producing lots of internal heat and possibly regulating that heat production, but also regulating pathways of heat loss to be the temperature that you want to be. Versus being an ectotherm, which means that your body temperature reflects much more your sort of local microclimates and the where you situate yourself in the environment. And so regulation comes down to moving through microclimatic mosaics in the environment as a way of a regulation. So let's just focus on endothermy and maybe give us just a sort of the physiologist overview of what endotherms do to regulate body temperature.
So the classic sort of Schollander-Irving model of endothermy is if an animal's keeping its body temperature, we'll say constant, say 40 degrees, then as ambient temperature slowly increases, say from 10 degrees up to 30, you have a decrease in the metabolic heat production that's required to keep that body temperature constant. And the slope of that line relating metabolic rate or metabolic heat production to ambient temperature is the thermal conductance, which is a measure of how easy it is to release heat to the environment. So if body temperature is staying constant, you increase your heat production as it gets colder, you decrease as it gets warmer, and eventually you'll get to the thermoneutral zone, which is where you're in thermal equilibrium. So you don't have to do anything active to stay warm or cool. Yeah. Once you get to ambient temperatures above that, the upper critical temperature, then you are saying to need to do something to keep cool. So you actively have to cool. And when you get to the point that your body temperature is the same or as ambient temperature, or ambient temperature is even higher than body temperature, then the only way you can lose heat is through evaporative cooling.
Right.
So then you start to increase your evaporative water loss to keep cool.
Right. Awesome. So a couple of quantitative follow-up questions about that. So, I guess my human intuition is that if I'm out on a cold day, my metabolic rate goes up because I'm trying to stay in my thermoneutral zone. I'm maybe shivering, but how large are these increases in the cold end of the thermoneutral zone that animals have in order to stay in equilibrium?
So once you get below the thermoneutral zone, then you have to increase heat, and heat production is going to be less than about 10 times basal. So that's going to be the really extreme end of how much you can increase your metabolic rate. So it's usually a fair bit less than that. So 10 times is getting to the top end of that.
But even
range.
2 or 3-fold sounds like a lot, right? I mean,
yes, yeah. So so you're using a lot of energy to stay warm. So there's things you can do. So you can decrease your thermal conductance. So warm coat, increase blubber. There's things you can do to reduce your heat loss, so you can reduce that energy. But those things aren't necessarily changeable. So if you're living in a varied environment, having a big warm coat is not something you can instantly change. So there's a balance between how you can moderate that from time to time versus whether it's sort of fixed. And that's going to depend on the environment that you're in. So some really well insulated animals live in really warm environments. They have thermoneutral zones that drop right down to 10 degrees. So they can.
So at the other end, if we get to this hot end of the thermoneutral zone and they're starting to go beyond that, you said that the only way to keep themselves cool enough is by evaporative cooling. But is it also true that metabolic rates go up once you get above the hot end of the thermoneutral zone?
Yes, so there's a point where you are no longer able to keep your body temperature at what we call normothermia. So you are going to get an increase in body temperature. So your cooling mechanism's not enough to dissipate all your metabolic heat production. And that's when you start to get an increase in body temperature. And then because of the Q10 effect, you'll get an increase in metabolic rate that corresponds to that.
I see.
And some of the energetic some of the heat loss mechanisms like panting can have an energetic cost. So you might see an increase in metabolic rate because of that.
And so that can be sort of a runaway process, right? Because the increased metabolism itself is creating more heat, which is the exact problem that they're trying to deal with. And so, can they get into this zone where it's just impossible to control because metabolic rates are rising?
Yes, eventually you get to a temperature, an ambient temperature, where you just can't regulate body temperature anymore.
Yeah.
You can increase your evaporative heat loss. Some animals will let their body temperature go up. So if you can increase your body temperature, then you're keeping that gradient there for conductive and convective heat loss. But there's eventually a point where you just can't do it anymore. And then you've got to trade off whether you worry about dehydration from losing too much water or whether you worry about becoming hypothermic.
Cooking in your own juices.
So vivid. Christine, you said Q10 just a minute ago, and I wonder that might come up again later. Can you define that?
Yes, so that is the change in a rate for a 10-degree change in temperature. So if you increase your body temperature by 10 degrees, how much is that going to change your metabolic rate?
Right. Okay. That's a really nice way of sort of thinking about standard amounts of change in different physiological systems and functions and things. So, yeah. In case
most biological systems, it's about 2 to 3 times.
Okay. So, all right. Good deal.
Yeah, so which means the rate changes by 2-fold or 3-fold with a 10-degree change in temperature. Yeah, just to be pedantic.
Close it all up. Yeah. Good deal. So we did a little bit about the what. We're going to talk a lot more about the what when we get to the specific avian research that you were doing. But why? Why did endothermy evolve? What's the thinking? I mean, presumably ectothermy came first. I am implying that, but why did endothermy evolve?
I think there's a niche there. So neither endothermy or ectothermy is better or worse than the other, but it does enable animals to exploit niches that are not necessarily ideal for ectotherms. So nighttime when it's cold, high altitude, there are niches to do things. And it means that you're more often in your optimal temperature. So in theory, you can reproduce more efficiently, you can get food more efficiently, but there's a cost, and that's the energetic cost that you pay. So endotherms have higher metabolic rates, even for the same body temperature, because I've got to keep all that metabolic machinery ticking over. So there's an energetic cost, but the advantage is that you're optimal most of the time, and you can exploit niches that you might not be able to exploit if you're an ectotherm.
Do we know how many times it evolved? Was it a single evolutionary event, or? Yeah, that's one of those fun questions.
That's a good answer. I like
Exactly 7.
I mean, there's endothermic invertebrates, like bees are a good example. Incubating female pythons are another classic example of endothermic ectotherms. And then you have your endotherms that are endothermic some of the time. So a torpid mammal, for example, just thermoconforms some of the time. So the line isn't clear, but endotherms have the capacity to regulate their temperature within.
That wasn't quite a fair question because there's all different diversity within endothermy. So yeah, to be fair. Good. There was another question I had as an extension. Now it's just blanked on it.
Well, I want to ask about of the major groups that have endothermy regularly, those include mammals and birds, and yet they have different temperatures to which they regulate in general. Birds are hotter. So why are birds hotter?
Mammals and birds have independently evolved endothermy, so it didn't come from the same place. So they've independently gotten there, and they just have come up with different body temperature set points. Although the difference may not be quite as much as we think, because birds are diurnal, so their highest body temperatures through the day and their lower body temperatures at night, and most mammals are the opposite. And so it's convenient to measure birds through the day, and it's convenient to measure mammals through the day. So you're measuring one in its rest. So resting body temperatures for birds is sort of 38 degrees for most birds, and they're 42 when they're running around. So yes, birds are hotter, but they may not be quite so much hotter. So, resting mammals typically 37 degrees, the resting birds 38, maybe 39.
Mm-hmm.
And mammals, when they're running around doing stuff, can be up to 40 degrees. So there is a difference. Birds are hotter, but there's a bit of an artifact for measurement and convenience of measurement as well. So if you work on birds...
I mean, it sounds like you're invoking just sort of historical happenstance to explain it, but are there any adaptive arguments for why birds might be a degree or two hotter than mammals?
I think the reason they are is just an accident. It's just an evolutionary accident, but it does bring with it some advantages. So if you're a bird and you're out in the sun, your body temperature is going to be higher than ambient temperature to a greater extent. So you don't have to worry about resorting to evaporative cooling so soon. So there are some ecological advantages. The cost of that is you're running hotter, so it's energetically more costly. So there's advantages and disadvantages, and that is going to be different in different ecological contexts.
Right.
It has nothing to do with flight, right? I mean, bats are not appreciably hotter than mammals, as I recall.
No, not well. They're one of the warmer mammals. They're certainly not a marsupial, but they're not crazy hot, but they're up there.
Okay.
This issue of hyperthermia as a way of dealing with arid, hot environments is pretty interesting too. And I just there are a few classic examples of mammals that live in large mammals that live in hot areas that let their body temperatures rise very high, right? So, and that's a mechanism for avoiding having to spend all of the water to keep your body down in the sort of normal human zone, right? So how hot are the hottest mammal bodies out there?
Usually about 46 is lethal. So
46.
Once their body temperature's getting up to 45, 46, then that's generally lethal. So most mammals, 43 is a bit of a problem. So when they're running around active, 40, 41 maybe, but once they're getting to 43, 44, 45.
That's pretty hot.
Pretty hot, they struggle.
I'm just thinking of how I felt with my hottest fevers in my life, and it's miserable, right? I mean, there's other reasons to feel bad besides just being hot.
For birds, I think Andrew McKechnie measured body temperatures of 48, 49 for, I think, Quelea. So there's some of the highest temperatures for endotherms, whereas your ants can get up to 55. There's some of those really hot ants. So the endotherms aren't quite up there, but 49 is pretty hot for a bird.
Yeah.
So,
that's just amazing. I mean, that's such an incredibly warm, and you hear all the time. You talked about the penalties of high body temperature being water loss, which deserts of Australia, definitely a big problem. But what's the temperature where we start to worry about proteins breaking apart and enzymes not really working? I mean, is that in a different range of temperatures, or is that part
Yeah,
of
so
what's
that's
going on here?
getting up there, and it's quite interesting that there is that consistency. So, for most endotherms, it is 45, 46 that's problematic for most things. And yeah, that's probably got something to do with enzymes starting to denature. And if you were going to deal with them when you're at lower temperatures, then they wouldn't work properly either.
Right.
So yeah, that is problematic. But it's the nervous system that seems to go to pieces first. And that is sort of the immediate cause, but yeah, ultimately the enzymes will become denatured.
So is it known mechanistically what about the nervous system fails at high temperatures?
I'm not sure. No.
Okay.
Oh.
Neat.
Art, you had an ectotherm question? We've always got to circle back to insects. We're not allowed to talk only about vertebrates on the show.
What was you ask it, Marty? What was my ectotherm?
Well,
question?
I'm not gonna channel it appropriately. It's the difference between you're asking about birds and performance, or I think you.
Yeah. So here, okay, let me run a pet hypothesis by you here. So
this is fun. We do this a lot.
Just run my random ideas by our guests.
Destroy Art. This is called the Destroy Art's Ideas segment of the show.
Well, so I think a lot about Q10 and the effects of temperature on metabolic rates and activity, because I come from an insect perspective, and generally performance goes up quite a bit as insect body temperature rises until you get to that maximum point, and then it falls off very fast. And so my sort of pet hypothesis about birds and mammals is that in a sense, there's also a Q10 effect, right? Birds and mammals are on some kind of meta performance curve for all the things they do. And I was wondering if birds are hotter as a way of just gaining a slight performance advantage. All of their systems are running faster. They're perceiving the world faster. They're just able to react slightly faster by virtue of being a couple of degrees hotter, and that maybe in the aerial environments that they largely occupy, maybe that's an advantage. So what do you think of that idea?
Yeah, it could be if you can afford to pay the cost.
Yeah.
So if you can pay that energetic cost, then yeah, you probably have that slight.
But maybe they can, right? By virtue of being aerial and being able to cover very large
dimensions.
Essentially, they have access to resources in theory that maybe small, ground-dwelling mammals do not.
Yeah, it's certainly a possibility.
I'll take that as no. That's not a good hypothesis.
No, hey, you weren't totally shot down. I would be happy about that. So, Christine, can I give you one that let's play the shoot Marty down part of the show. I have been completely enamored by the concept of fever since I started to be formally trained in biology, and I think the prevailing argument right now is that fevers are around to sort of get the immune system, parts of it at least, into a higher idle for when the bad guys are around, because especially bacteria tend to be really sensitive to temperatures that we can endure. I mean, that's not actually the main point. The question really is, do we know of, or do you think that upregulated set points of body temperature, without going into the details, that's actually what fever is. Do you think that is something that may be more pervasive than we really recognize? Because for sure, you're talking about torpor, you're talking about playing around with energy savings by allowing body temperature to change. I just wonder if there are something like fever protecting the body from infection. Are there other changes in body temperature set point that sort of are these short-term improvements or facilitations of other kinds of performance that just haven't been tackled before? Is there anything out there like that I've missed, or is that crazy?
Yeah, I mean, I think now with more and more sort of onboard data logging, we're starting to see those natural fever events. So, high temperatures associated with infection or reproduction and those sorts of things that we're starting to come through. So, some of the work coming out of South Africa, where they've got data loggers in some of these big mammals, and they can say, okay, the animal gave birth on this day, we can see it from the body temperature trace, or this animal had an infection, there was a fever this day. So I think that those data are starting to come out more and more now that we can monitor and log animals in the field longer term as the technology is getting better and better. So I think we will see more and more of those data coming out.
Cool.
Marty, are you implying that birds have somewhat higher body temperatures in general because they're exposed to pathogens more regularly, and this is a mechanism for dealing with that?
multiple
where you're
people
going?
have invoked that. Yeah,
Huh.
but that's a possibility. Yeah, well, I don't want to go too far down that road, but people have speculated that. And I did some work as a graduate student on this genus of mice called Paramuscus, and their idling body temperatures tend to be higher than a lot of other small mammals. So we invoke that. I mean, that work was specifically immunological, and there were other elements that gave credibility to it. But yeah, that's a part
Cool.
of where I was coming from.
Cool.
But let's talk more about your specific work, Christine, and in particular, this paper that you had not too long ago in Journal of Experimental Biology with Laura Hurley and Pierre Deviche and Simon Griffith. And what you were trying to do there was get at what we've been talking about, this issue of climate predictability. I think you were. If I'm misinterpreting, you tell me that I'm off. You're interested in climate predictability. You were asking about what exposure to high and low temperatures did to energy metabolism and body temperature in one of the iconic species, bird species of Australia, at least one of the ones that's in our pet stores here in the US, the zebra finch. And you were wondering about what happened to the ability of those birds to regulate body temperature and run their metabolism generally when you're exposed to short-term changes in temperature after having been experiencing these long-term conditions. So in a sense, that's all wrapped up in your words in this sort of emphasis on physiological plasticity. I think to be able to talk about the rest of it, given that you frame your paper that way, is probably how you want to do it. Can you tell us what you meant by physiological plasticity and why it in particular is meaningful with respect to this extreme unpredictability we've been talking about?
Yep. So the physiology of species isn't set and fixed. So we know it can vary. So if you measure, the basal metabolic rate of a bird in summer or winter, it's going to be different. If you measure the lower critical temperature, the upper critical temperature, it varies. So we know that birds and mammals can respond to different environmental conditions, and they do so within their current physiological scope. And so with this study, we were really interested to see how they could modify their physiology to better cope with these extreme events. So we'd done some earlier work measuring energy and water turnover in the field for these zebra finches in hot and cold periods. And their water turnover didn't change, their body mass didn't change. So it was like, well, what are the mechanisms that they're actually using to deal with this? How are they keeping, maintaining body mass when it's cold or when it's hot? And why aren't they really changing their water turnover? So we wanted to see what the underlying physiological mechanism was. What could they change about their physiology to allow them to accommodate these different conditions?
Mm-hmm. Sorry. Coffee in the wrong pipe. I wanted to ask a follow-up question about just the philosophy of thinking about plasticity. And that's where do you draw the line, or do you draw a line between plasticity and just physiological flexibility and changes in just sort of very short-term physiological systems, right? So if I go out for a run around the block, my heart rate will go up. Is that plasticity, or are you talking about something different when you talk about plasticity?
So I guess it's a repeatable response to a certain condition. So seasonal is sort of the classic example. So it's repeatable. If you measure a bird in winter, it's going to change. So if you've got some baseline value, does it change either way for over some sort of time period? And how quickly they can respond is a bit of a question. I mean, in most of the lab work, people acclimate their birds. They bring them in. They put one group in, in a room at 15 degrees for 5 or 6 weeks. They put another group in a room at 35 degrees for 5 or 6 weeks, and that's sort of what we think about as plasticity. But we were really interested on how do they do it, over 3 or 4 days. And we waited 3 or 4 days before we actually measured the birds. It would be really interesting to go back and do it after the first day of a heat wave, the second day of a heat wave, the third day of a heat wave, and see how quickly they're changing in that respect.
But you think like minutes to hours, that's too short for that really to be meaningful plasticity. That's something else.
That's sort of your aerobic scope. So if you go for a run, how much can you increase your metabolism, to accommodate your demand at the time?
Okay, so somewhere in there, there's a line between those two kinds of responses, and is it possible to identify that line?
I guess when we think of plasticity, we're thinking about sort of the whole metabolic profile. So you could measure maximum metabolic rate after acclimation or acclimatization to certain conditions. So it's looking at, if you measured sort of the whole, if we think about the Schollander-Irving model, that metabolic profile over a range of temperatures, you could see that pattern still there, as opposed to, if you go for a run, you're increasing your metabolic rate, and that scope could always be the same, or it could be plastic, it could change. So you're looking at that whole change of temperatures and where does it sit on the sort of ambient temperature scale.
I see.
So it sounds like, and I think a lot of people would agree with you, that the qualifier for plasticity is that you have a repeatability in the response, right? And I think the reason that we care about the repeatability is that that allows us to think about plasticity as a trait. And it's a trait, meaning that natural selection can act on it, and therefore it can evolve. And so, what is that window, over what time scale, what context would something switch from being flexibility, to use a very generic word, to being plasticity? That may be one of the biggest things that biology has to wrap its head around. In fact, haven't we done 3 or 4 shows on that? So it would be a lot to expect you to get that. But I think, that piece about where, are traits repeatable? Are these plasticities repeatable in the window of time over which you get it? Do you think that oh, sorry, go ahead.
And it's something we really need to consider. So when we do sort of mechanistic distribution models, people plug in, the metabolic rate of a species, the water loss of a species, the body temperature, but there's certainly scope for that species to modify that, and that's often not considered. So I think there's a lot of scope potentially for species to cope with more extreme environments or changes just within their existing physiological scope. Adaption's there, but it's a slower process. But if species can shift their energy and water requirements, then there's scope for them to accommodate.
Wasn't there some work done not too long ago? Maybe it was by McKechnie and others, asking about relationships between, say, basal metabolic rate and metabolic scope. I mean, there are even relationships there between the foundations and the plasticity, right? Do you remember how that went?
Different authors find different relationships, so it sort of depends a bit on the data that you use. But generally we assume that your field metabolic rate is about 3 times your basal metabolic rate. For some taxa, it works better than others. Your metabolic scope, it's not going to be much more than 10. It's usually 5, 6 to 7. But again, it varies a bit with taxa and as to the scaling relationships that are there.
Okay. Great. Let's circle back to zebra finches. We kind of got diverted off into philosophy again, which is fine. We do that all the time. But let's just talk for a minute about why did you choose zebra finches as your focal species, and what did you do? It sounds like from the paper it was some pretty heroic field work and making some pretty sophisticated measurements in the field, which I'm always super impressed with by physiologists. So tell us what's up.
We've got a study site out at Fowler's Gap, which is out in Central Australia near Broken Hill. And Simon has had nest boxes out there and been studying finches out there for a long time. So they're a really good population to study because there's a resident group of birds that are there. We put in some artificial water so we could keep a small number of them there. And they're an iconic species, so they're a desert species, but they're also a species that we know have been susceptible to some of these mass die-off events. So how and if they were dealing with these high temperatures was really important. And Fowler's Gap was a great place to do it because it's a university research station, so there's power and a lab available to do that work. But yeah, it was pretty hot when we were out there, which was great. When we applied for a grant to study birds in heat waves, which seemed like a really good idea at the time, and so we realised
so you have to do the work.
We were in the field in the heat wave.
So how hot are we talking during the working days?
The maximum I think we got to while we were there was about 46 degrees Celsius.
Wow,
so,
brutal. I've worked in Arizona in the low 40s, but not really in 46, 47. That's hot.
Yeah, it was pretty hot. So, and the air conditioner used to die once it got over to about 33. So it would work fine below 33, but once it got to above 33 in the lab, the air conditioner used to pack it in.
Can you say more about how you've done this? We don't usually get into the gory details of methods in these talks, but this is pretty cool how one gets data about body temperature and especially metabolic rate from little animals like this. So you're going out to these brutally hot places in the middle of nowhere, you're capturing these birds, and then what? How are you getting the data that we're going to talk about in a minute?
So there were sort of 3 parts to it. So the first was measuring their field metabolic rate. So that involved catching the birds, giving them an injection of isotopes of oxygen and hydrogen, taking a blood sample, letting them go, catching them 2 days later, taking another blood sample, and then looking at the turnover of the isotopes. Then we wanted to look at the mechanistic side. So we wanted to measure their metabolic rate and water loss. So that was open flow respirometry. So we dragged out a couple of respirometry systems, set them up in the lab there. We'd catch the birds in the morning, we'd fast them through the day in an outdoor cage where they had water but no food, and then we'd measure them overnight. And then we'd also catch some birds to do some hormone blood chemistry.
Okay.
So for the uninitiated, that's two different ways of measuring metabolic rate, right? So can you say what those are?
So field metabolic rate is measuring the energy of a wild free-living bird and the water turnover of those birds while they're out there doing their thing.
And that's what the isotopes allow you to do,
that's the
right?
isotopes allow us to do.
Okay.
Yep. And then the open flow respirometry is a standardized measure. So you can put your animal under standardized conditions. So we did them at 30 degrees, which is thermoneutral, and 40 degrees, which is a tolerable but challenging temperature for these birds. And then you can measure their metabolic rate, their water loss, and you can calculate their conductance, and we measure their body temperature as well. So one's a sort of standardized approach using really standard techniques of a resting bird under controlled conditions, and the other is what a bird is doing. It's sort of a snapshot in time of its energy and water turnover.
Yeah, gotcha.
Yeah, and the process of doing the respirometry it was one of my favorite techniques. Frustrating because of all the little things that can go wrong, of course, but one of my favorite techniques that I used to do a lot in graduate school, because you have, the animal in some sort of vessel where you can control the air going in and out, and you're measuring the sort of consumption of oxygen and the production of carbon dioxide. I think you didn't do the latter part, if I remember, not that it really matters very much, but that whole setup and the various pumps that are involved. And I mean, this is a, completely humane process. It's all done in the dark. And if you turn the lights off for birds, the vast majority of them, they just calm down and there's no big deal. But the process of doing that and seeing the setup, I know for graduate students and well, anybody that I would ever show that to, they're like, what is this? So it's
looks
it's
like
just
mad scientist setup, right?
Yeah, I mean, that's the quintessential mad scientist setup right there.
There's no bubbling green stuff, but otherwise,
no, you put that on the side, just to make everybody happy. But yeah, it's really cool. So the two major findings that I remember from the paper is, surprisingly, that hot days were not stressors for the finches in terms of the birds maintaining their body mass as long as you were giving them drinking water, and that they also shifted their thermal neutral zone to these higher temperatures. So let's do these in parts. They're not stressors? These birds are not experiencing these 40 plus degree days as stressful?
No, so they were able to accommodate them. So they didn't lose body mass. In fact, we found that the cooler days, and cool out there was anything below about 36, they were more stressful for the birds. They actually lost a bit of body mass on the cooler days because they'd had a drought for 2 years. There'd been no more than about 10 mils of rain events in the last 2 years. So there was very little food around. And so I think the energetic issues when it was cold and having to keep that heat production up was worse for them than being able to accommodate those hot days. So they could do it. And that's why we're interested in how did they do that. So when we put them in the lab and measured their physiology, so they were letting their body temperature go up by about 3 degrees. And that meant they were saving water. And that water temperature trade-off was probably giving them about 3 hours extra that they didn't have to drink through the day.
That's amazing. I don't know how much people know about zebra finches, but to add the details to the system, it makes it stick even more for me. These are very tiny birds. They're 15 grams or so on average. Is that about right? Even smaller than that, right?
15. Our field ones were 11 to 12 grams.
Okay.
So
and
some
then
of the
we're
insects
talking
I
about,
worked on
again,
are bigger than that.
Yeah, exactly. And the temperatures, for the Americans that are listening, we keep throwing around 40s and these kinds of things. 42.7, which is, I think, some of the temperatures you were at some point exposing them to, that's 109 Fahrenheit. So that's tiny bird, 15 grams at 109 Fahrenheit, and they're brushing it off. Don't bring the cold temperatures to me. These warm temperatures are no big deal.
Yeah.
Yes,
So,
and they seem to be doing that a lot by predicting the hot days. So days that were really hot, they were coming in early to eat and drink. They were disappearing through the middle of the day, and then they were topping up in the afternoons. Whereas on the cooler days, they were eating and drinking throughout the day. So I think it was that predictability of, it's going to be hot today, I need to get my food and water early, was letting them get through the day. So they were somehow knowing, and presumably from the early morning temperatures, that it was going to be hot that day, and they were anticipating that. So before it got hot, they were feeding a lot more in the mornings.
Wow, interesting. So let's also talk about this idea of shifting your thermoneutral zones and the higher equilibrium body temperatures of the finches that you found, right? So I think you said in passing that they were 3 or 4 degrees warmer in the hot treatments, right? So how do they do that?
So during the hot phase, you've sort of got two ways you can shift your thermoneutral zone on the short term. So you can either change your conductance or you can move your body temperature set points. So it seemed that these guys were just moving their body temperature set point up. And so that equilibration temperature was at a higher ambient temperature. So you can't easily, acutely change your conductance. You can do it to a certain extent, but once you get to those high temperatures, you're already maximizing your thermal conductance. So they can't suddenly shed all their feathers and molt and grow new ones. So the most logical thing they were doing is changing that body temperature set point. So if you move your body temperature up, then you're moving the ambient temperature at which you're at equilibrium up a little bit
gotcha.
as well.
And to be clear for our listeners, so when you say conductance, you're talking about the resistance to heat loss, or the inverse of the resistance to heat loss, right? So how easy is it for the heat to go out of the body?
Yes, so I think of conductance as the opposite of insulation.
Yeah.
So, yeah.
Okay, great.
Yeah, this is why the dropping the feathers would be a mechanism for conductance if such thing was a possibility. Okay.
Yeah, and you have wet and dry conductance. So your wet conductance includes your evaporative heat loss, and your dry conductance is just your non-evaporative heat loss, so convection conduction.
Got it. So in terms of so that shift in thermoneutral zone and body temperature, you would consider that a form of plasticity, right?
Mm-hmm.
Yeah. And in terms of just overall in birds, say stepping outside of this system, how common do you think these sort of shifts in thermoneutral zone and body temperature are as a way of dealing with hot temperatures? Is that
for desert birds? I'm guessing that they're reasonably common. So we know that they can acclimate and acclimatize. So we know they change seasonally. We know they change in the lab. But there hasn't been a lot of field work done. So there's been a lot of really high temperature field work that Andrew McKechnie's been doing lately, and he's showing some acclimation from different sites. And this was really interesting to do it really acutely. So there's been some sort of more chronic acclimation and acclimatization work done. But yeah, it was interesting that I could do it so acutely. And as I said, the next step would be really nice to go back and see, what happened after 1 day of a heat wave, what happened after 2 days, what happened after 3 days. So we measured them at the end of the third day.
Great.
And again, just for the listeners, I think you're saying acclimation and acclimatization. I don't know if we really want to get into the details of what distinguishes those, because it's kind of esoteric for everybody except those of us that do this kind of thing. But the acclimation is this sort of longer scale adjustments, presumably involving some of the same short-term plasticities that you're focusing on. Is that right?
Yeah, usually we use acclimatization for wild animals in the field and acclimation for captive animals in the lab.
Okay, but in general, it's just sort of prolonged periods of making adjustments to changes in temperature or
exposing
other
them
things.
to different conditions. Yeah.
Okay, good.
I wanted to ask also about mechanism, just the whole water loss issue. And you've written some really interesting things about water loss and regulation of water loss. And so, I think the human-centric way of thinking about this is sweating, right? So if we get hot, we start to sweat. But many things don't sweat, and they have other forms of or pathways by which they lose the water vapor that's carrying away the heat. So in birds, what are those pathways, and how do they exert control over them?
So for birds, the two major pathways is your respiratory water loss, so through the ventilatory system, or cutaneous water loss across the skin surface. So panting or gular fluttering is increasing your respiratory water loss when it gets really hot. But even at low temperatures, so thermoneutral and below, it's going to be some inevitable water loss across the lung surface and through the respiratory system. When it gets hot, they might not be able to sweat, but they can increase their cutaneous water loss. So they can increase the permeability of the skin. So at high temperatures, we call that sort of thermoregulatory water loss. And that's where they're increasing their water loss to lose heat. But at lower temperatures, there is inevitable water loss just because the skin is permeable to water vapor and the lung.
So that cutaneous water loss is really interesting, right? So birds don't have sweat glands like we do. But is the magnitude of the water loss on par with what we might see in a sweating human or a sweating mammal? Is it a big component of the overall water budget?
Evaporative water loss, so respiratory and cutaneous, can be up to 70% of the water budget.
70%
for some species, and partitioning water loss into respiratory and cutaneous is really tricky. So if you physically petition, so you stick a diaphragm around the bird's neck or you put a mask on, they tend to stress. And so then they hyperventilate, so then weird stuff happens to their water loss. So it's really hard to petition it non-invasively when the bird's calm and resting. So we try to model it, and different species vary, but 50-50 is sort of a typical petitioning. Some species you lean more to cutaneous, and I mean pigeons are weird, they have their weird cutaneous sort of sweating that's not really sweating, but for most birds it's probably 50-50, maybe a little bit more cutaneous and respiratory.
Interesting.
But petitioning's not trivial to do.
Yeah. And just an experimental thing. So you're talking about a collar around their neck, and by that you mean you would have them in a respirometry system, and you would partition the air that's going past the body from the air that's going past the head, so that you can tell where the water vapor is coming from, right?
Yeah. So some people have tried to do that. Some people have tried to put a mask on.
Right. And the birds just don't like it.
Yeah. I mean, you can train them so that they can be more accepting of it, but
Got it.
generally you find that there's higher water loss total if they're physically positioned. And so when we do these measurements, we really want, resting minimal rates that are really comparable, because if your animal's a bit awake or stirred up, then it's really hard to compare between species or between individuals, between conditions. So petitioning's hard, and that's why we've tried to mathematically model the petitioning, and I said it comes out about 50-50, maybe a bit more cutaneous.
Got it. In terms of total, sorry, Marty, one more question. So in terms of total,
just go ahead, Art.
Total water fluxes, like what percentage of their body might they turn over in terms of water every day? Are they really huge in these hot environments?
So the zebra finches were turning over about 4 mils of water a day. So they have,
4 mils out of 15 total grams.
Yeah.
Right? That's a lot. Wow.
That's a lot. Yeah.
Yeah. So that's when it's really hot. So they're obviously increasing their water loss. But of their total water budget, evaporative can be up to about 70% of their total water budget.
Wow.
Okay. Coming back to, shut up for a second. Coming back to the plasticity ideas, the difference between evaporative water loss by respiration versus the skin. I mean, the skin would seem to be one that was much less labile, right? So as far as the plasticity dimension has a role in the systems that interest you, I mean, is there much to put into cutaneous plasticity?
So there has been some work with birds demonstrating that they can change the lipids in their skin, so the composition of the lipids, but we don't know how quickly they can do that. So a lot of the acclimation studies have sort of been over weeks. There has been some old work on zebra finches showing they can change their skin composition over hours, like 16 hours. But we're finding that animals can actually regulate their insensible water loss, which is sort of the low temperature, inevitable water loss, over 8 to 10 hours. So we're not sure whether that's a combination of changing the skin properties or changing the respiratory or some combination of the two. But really the easiest way to do it is to change the temperature of the skin. So you change your blood flow. So that's sort of the obvious way and the quick way they could do it. But longer term, they certainly do change their skin lipids, but we're not sure how acute that response is.
The skin of birds is not super vascularized though, right? So how much option do they have there?
It's vascular enough they could certainly change the temperature by...
Okay. So I think we wanted to ask one more question. Was there anything else about this paper? I want to talk about parrots briefly, and you mentioned insensible evaporative water loss. So I do want to come to that. But Art, is there anything else about the?
No, I think
zebra finch paper
water loss is a good thing to turn to.
Yes. So tell us about that. I mean, the rate at which they can lose water across the skin, we've just been talking about that and the mechanisms by which they can do it. But in other species, especially in species that live in fairly wet habitats, it seems to be something that they may be surprisingly adept at. Tell us about this other JAB paper on the two parrots.
Yeah, so a lot of the work on water loss is focused on thermoregulatory water loss. So what's happening to water loss at high temperatures when animals are losing water to lose heat. But there's always water loss happening. So at thermal neutrality and below, animals are losing water just because their skin and their respiratory surfaces are permeable. And that's what we term insensible water loss. And for a long time, we thought that was just physically driven. So it was dependent on the water vapor pressure gradient between the animal and the environment. So if it was really humid, you would have a smaller water vapor pressure gradient, so you'd lose less water. If it was really dry, you'd lose more. But we've found that animals don't conform to this sort of physical model. So water loss, insensible water loss, seems to stay fairly constant regardless of what the water vapor pressure. So it's not that linear relationship that we've always thought there to be. And the obvious thought was, well, it's water conservation. So, animals are doing this so that when it's really dry, they're saving water. But we've found that mesic species seem to do it just as well as arid species. So that isn't really consistent with the hypothesis that it's for water conservation. So the other reason they might be doing this is for thermoregulation. So if you're at a given ambient temperature and you're at really high humidity, if your water loss is impeded, that means also your heat loss is going to be impeded. So if at any one ambient temperature, if you change the relative humidity of your environment and the water vapor pressure of your environment, that's going to impact on your heat balance, which means you're either going to have your body temperature change, or you're going to have to mess around with your heat production or your conductance to keep that body temperature constant. So if
Marty, are you there?
Uh-oh. Yeah, I'm here. Uh-oh.
We just lost her.
Oh, crap. Well, hopefully.
See if she comes back.
It's been lagging. I don't know. Are you seeing the video
That's not
lag?
lagging for me.
The audio is fine, but
thing.
there's a disconnect between the audio and video. Oh, crap.
Not for me.
Christine, I don't know if you can hear us, but obviously we can't hear or see you. Maybe if you want to log out and log back in.
Bail out and come back in. Yeah. Is the Zoom moment.
It's a minor miracle that we don't have 53 episodes and we've only had a couple of
more total disasters.
It's probably going to be 4 or 5 lightning strikes in a row here soon.
I know.
So I thought
hopefully not with Eric Jarvis.
Oh God, yeah. There's certain people we just can't do that for. That's going to be a heavy duty conversation. So see, I know you got a lot on your plate with it, but it's going to be a heavy duty one.
Okay.
Yeah. Read that paper carefully. It's short, but holy crap, there's a lot of meat to it. And I offered to do the bird sequencing things that he's been doing as in exchange, and they didn't respond. So we're just going to stick with the vocal learning initial invite. So when she comes back, I think we'll start move the wrap up section. Yeah.
Yeah, I think we need to talk a little bit about mechanisms of how they regulate the insensitive.
Yeah, I want to. Why is it called insensitive? Do you know?
Insensible. Yeah.
I mean, either way, what is that about?
No, we got to ask her.
You were worried. Okay, I thought it may be some obvious answer that I just
I don't think it is.
It's insensible. I mean, nonsensible. No.
Is
insensitive.
I think it's a relic of the way people talk about heat loss. But that's not the way they talk about it in their JEB paper. So, evaporative water loss is insensible, I think it's insensible heat loss, because the heat is in there. You're not measuring heat directly, right?
You're measuring it in the water.
Disappeared by causing the water molecules to evaporate.
Yeah. Okay. That would make sense. This is the heat that you can infer was there.
Yeah.
Yeah. All right. Well, I'd still like to validation that that's the real reason, but that makes sense.
Yeah.
It's such a weird word, such a weird term. Otherwise, what insensible? What?
I'm gonna check email here.
Yeah. I don't have one.
Nothing from her.
Mm-mm. Oh, sweet. I'm still recording, by the way.
Yeah, I am too.
Sorry about the coughing. I've been trying to hold it in. Sorry, especially to you, Matt. That's going to have to cut this stuff out. Oh, here she comes.
Hello, I think I'm back.
Good.
Hey, there you are.
Hey,
Sorry about that.
Right. I was just writing you a desperation email.
Yes, I was doing the same, but it seems to be back.
We figured you just had enough and decided to bag it, so we would understand.
These guys are idiots.
These guys
I'm
are
done.
jerks. So let's see. Do you want to? I don't know what's best for you. I can't remember exactly where in the ID.
Well, Christine, just to make sure you're still recording on your phone, right?
I just restarted it.
Excellent.
So.
Okay.
Good. Excellent.
Do you want to try to tackle that again? I think the problem is we don't have to go through the whole background. Go. What were you saying, Art?
Well, let's just pick up on where we were with insensitive evaporative water loss.
Yeah, if you remember exactly where you were, but sure.
Yeah.
Art and I had a conversation while you were trying to get back on about why it's called insensible. So now my head is stuck there.
Yeah. Why is it insensible?
It's just an old definition, and one of the things that I would suggest is that we do away with that term, because if our hypothesis is right and that they are regulating their insensible water loss to thermoregulate, then it's not different from thermoregulatory water loss.
Right.
So we perhaps should have water loss, above and below the thermoneutral zone, but you're really dividing it into insensible and sensible water loss is perhaps not as very useful anymore. But I think it's because we used to think one was regulated and the other was passive.
Got it.
But
Got it.
it seems to be that's not the case.
So if I can just recap, since we had a little break, recap what you said before. So you found in these parrots that live in quite wet human environments that they appear to have control over this sort of low level of background water loss. And the inference is that they're using that as a thermoregulatory mechanism. Is that right?
Yep.
Okay, great. So given that, how are they doing it? What are the mechanisms by which they're regulating?
So they could be regulating their cutaneous water loss, their respiratory water loss, or both. So if it was their cutaneous water loss, then their skin temperature is the obvious way to do it. So changing their blood flow, or they could be acutely changing their skin lipids, which is not impossible, but it's maybe less likely to be able to do it over.
Yeah, that's what you were saying earlier. Yeah.
Respiratory water loss, they could be changing the temperature of their expired air. So we know lots of animals have nasal countercurrent heat exchange systems, so they can cool the air that they're breathing out. So that's one way they could reduce the water content, or perhaps they're actually expiring air that's not saturated. So we always assume that animals expire air that's 100% saturated. There are some species, so camels, ostrich, that don't necessarily expire saturated air. So perhaps the air is not saturated. So that's sort of the next avenue, is to have a look at what mechanism are they using.
How saturated is the air that we expire? Is it 100%?
Pretty close to 100%, yeah.
And that's because we have not very nice nasal turbinates and we can't depress the water.
No control.
Yeah.
This is one of our less impressive traits. Yeah. Yeah. I've always been jealous about species with nasal turbinates, and I wish I had better ones.
We need to do a whole episode on why humans aren't special. Just list off all the traits. How humans suck. Christine, so thank you so much for telling us about all the work that you've done. Not all the work, obviously, but at least a couple of flavors. But we're interested here about what's coming next, and especially in the arena of physiological plasticity.
So at the moment, I'm really interested in whether animals are more resilient than they are supposed to be to climate change and extreme events. So I'm sort of halfway through a project looking at echidna heat loss. So echidna is another one of those species that are meant to really struggle with high temperatures. Mm-hmm. So, their lethal temperatures and ambient temperatures are meant to be about 38 degrees, but we know they're out there running around when it's 38, 39, 40 degrees, and they're certainly not dropping dead. So we've been doing some thermal imaging work to look at their potential avenues of heat loss. I'd really like to do some work on desert parrots. So sort of to continue the zebra finch work onto some sort of common desert parrot species as models, and then get into some of the more threatened desert parrots like night parrots and princess parrots.
I mean, is the echidna sort of an arbitrary choice, or what is the motivation? Is there something special about the echidna that makes it especially sensitive, or
it's
notorious for its sensitivity? Yeah, see, that's what I'm saying. It's an echidna, which makes it stand out. But was there something beyond weirdo, spiky-looking mammal creature that motivated its
studies? Echidnas are really interesting. They're monotremes, so they have a really low body temperature, really low metabolic rate, and theoretically a low heat tolerance. And it's always been argued as to whether this is adaptive or whether it's primitive. Is it just that they're a monitoring or a primitive mammal, so they're hopeless at thermoregulation, or is it adaptive because they eat termites and ants, which is a pretty lousy diet, so they have a low energy lifestyle, so they do everything low and slowly. So this has been and I'm particularly interested in myrmecophagous. So I did my PhD on numbats. I've worked for a long time on echidnas. And so sort of solving that question about, if they are more tolerant to heat than what they're supposed to be, what's the mechanism? How do they do that?
And I guess platypus, the only other monitoring we have, we don't worry about that so much because it's mostly aquatic.
Yes. So they obviously have quite a different heat issue. So heat loss is probably more of a problem for them in aquatic environments.
Yeah. So if you look forward 5 or 10 years, are there oncoming technologies that you view as being transformative, and are there kind of fundamentally new things you're going to be able to measure and do?
I'm really interested in small species and their interactions with the environment. So the technology to have smaller and smaller loggers to get field data is really exciting. So batteries are the sort of the limiting factor at the moment. So for loggers and transmitters, then you're trading off battery size against battery life against range.
Right.
So as we can get these things smaller and smaller and smaller, we can get them into smaller and smaller animals, which are the ones that are thermally the most interesting because they're more variable. So that to me is really exciting. And we've already got these little tiny temperature pits now that we can get into a zebra finch. So
yeah,
it's going to get really exciting.
Yeah, so to be clear, these are mostly data loggers that you're putting inside animals. They're either swallowing them or you're implanting them surgically. Is that right?
Yeah, so we usually surgically implant animals into the abdominal cavity.
And those are recording body temperatures and then transmitting them to...
Yeah, so there's different approaches. So one's the logger approach, where it just is onboard logging, just collects the data and you have to retrieve the logger. Then you've got transmitters that give a signal and you record that the beat rate. And then there's pits where you have a receiver pad, a bit like the pit you put in your dog for the ID. And that can also do temperature and activity. So if you've got a receiver pad sitting there, the receiver pad actually energizes the pit. So you don't have to worry about
it's
battery
passive, so there's no battery. Yeah, got it.
Yeah. So there's sort of three different...
Sorry, go ahead. I'm just curious about these ones at zebra finches for my interest in fever that I was talking about before. So this is almost offline of the podcast. Which one of the three are those?
So these are the ones with the receiver pad. So they're pits. So they don't have, so they can be really small because it's the receiver pad.
Right, and no battery. How close do the birds have to be to the pad for the activation to happen?
Probably 10, 15 centimeters.
Okay, pretty close. Yeah.
Yeah. So I mean, for water in the field, you essentially net your water source and have the birds have to pass through the antenna.
Gotcha. Okay. Yeah.
And in the lab, you sit your chamber on top of it. But yeah, they're getting smaller and smaller, which is really exciting because we can start to do more.
That opens up all sorts of doors. Yeah, absolutely.
And for longer term studies, so you can monitor them over seasons, and I mean, heat waves are unpredictable, so you're never quite sure when one comes. So the longer that you can collect these data, for, the more
likely
exciting.
to get what you want. Yeah. So we've been asking you a lot of questions. One might say grilling you. What else would you like to say? Would you want to make sure that the audience knows in terms of climate change, bird energetics? What else did we not get to?
I guess how exciting it is to do science and be able to tackle some of these big questions about the environment and how animals respond, and to be out there, and the importance of a combination of, I guess, lab and field approaches. So lab approaches tell you what animals can do, field approaches tell you what they do, and the combination of those two things I think is really powerful for looking at what are they doing and how are they doing that. And sort of the utility of some of the pure science to potentially have applied outcomes down the track. So, we might look at how animals regulate their water loss, which is not necessarily an applied question, but we need to know how animals work to be able to then apply that to conservation and management down the track. So,
spoken like George Bartholomew, almost.
Yeah. Perfect.
Great. Well, thank you so much. This was a lot of fun. Sorry about the little technical challenge there. I appreciate you coming back.
All right.
And we can truly blend it out. Nobody will ever know that it happens. The one thing that I'll ask you to do whenever you can, just send us the file by whatever mechanism is easy for you, and we'll let you know that it comes and confirm that it's all okay. Anything else operational?
Well, so once we get to the closer to the production and release date, we'll ask you for a headshot, and maybe you already said that, Marty.
No, I didn't.
And we'll just kind of circle back around and coordinate on social media pushes.
And not just a headshot, but we appreciate other pictures of you doing the field work and zebra finches outdoors and all those kinds of things for social media and such.
Echidnas.
Yeah. I was going to say even echidnas, but then it's like a false advertising because we don't talk about echidnas, but briefly at the end.
But we don't want one of your husband's pictures. We want one of yours.
That's right. Excellent. A good deal. Thank you so much, Christine.
Right,
we appreciate it.
Thank you for having me.
Yeah,
absolutely.
absolutely. So hope to get to meet you in person at some point.
Yes, eventually we'll be allowed out of here.
One day we can do this.
Yes.
All right, great. Take care. Thank you so much.
See you later.
See you.
Okay,
Bye.
bye-bye.